Bipolar disorder, also called manic depression, is a neurological disease that affects between 1 and 3% of the world's population. It's categorized by extreme shifts in mood to the point where it impacts an individual's ability to lose interest in activities, feel hopeless, and have difficulties concentrating on everyday tasks. Individuals with this disorder can experience troubles with performing work-related tasks and interpersonal activities. They also have a higher rate of being jailed, arrested, and convicted of crimes than those without a diagnosis. It's critical for scientists to understand bipolar disorder to find effective treatments. And the best way to do this is by using an animal model that can replicate the human phenotype. A common symptom of bipolar disorder is a disruption of circadian rhythms and sleep habits of the individual. Often people that are affected by bipolar disorder are plagued with bouts of rash decision-making and risky action. In our experiment, we had 4 different treatment groups, the first being ketamine, which induces risk-taking behaviors, in which we injected the ketamine into the rats, and it also reverses depressive behavior. The group was HCA, which is homeocystic acid. It induces a depressive state, and we injected this into the rats. Our third one was ketamine and HCA. We injected this into the rats, and it's a representative of both manic and depressive states. Finally, we had our control group, which was just a saline injection.
For this project, our research question was, can HCA injected into a rat induce a manic depressive phenotype? And can pre-treating with ketamine prevent this phenotype in animals injected with HCA? Our overall hypothesis for this experiment was that rats treated with ketamine would exhibit lower anxiety than any other group, given that our class was specifically testing the ketamine group of rats. In our experiment that we did, we had 4 groups of rats that were treated. We had our ketamine-only rat group, our HCA injected rat group, our rat group that was injected with both HCA and ketamine, and our control group.
For the open field test, each rat was individually placed inside a 100 centimeter by 100 centimeter by a 40 centimeter open plywood box in an isolated room, while a computer program analyzed the box in sections and recorded the rat's movements throughout a period of 5 minutes. They specifically tracked how long the rat spent in the 50 centimeter by 50 centimeter center portion of the box and how long they spent closer to the walls. The elevated plus maze consisted of a contraption of 4 arms facing each direction 90 degrees apart. 2 arms were open with no walls, and the 2 opposite arms were enclosed with walls on each side so the rats could not fall off. Each arm was 50 centimeters long and 10 centimeters wide, and the walls were 30 centimeters high on the 2 with walls. The entire plus was placed on 2 sturdy legs standing 50 centimeters off the ground. Each rat was placed individually in the middle of the plus facing an open arm oriented toward the back of the isolated room. A software computer program and camera placed 4 feet above the maze was again used to track the rat's movement. The video tracking system divided the maze into 3 zones: the very center of the elevated plus maze, the 2 open arms of the elevated plus maze, and a second closed arm of the elevated plus maze. Each rat was allowed exploration for 5 minutes without human interference. The computer tracking program recorded the number of entries the rat made to each open arm versus the closed arms, the amount of time spent in the open versus the closed arm, and the total distance traveled by the animals. The third test was the Morse water maze. This test required a computer tracking system and camera placed approximately 4 feet above the maze, a watertight pool, 150 centimeters in diameter, and room temperature water that had a small concentration of black tempera paint in it so that the platform was not visible in the water solution. Enough water was added to the pool so that the rats could not touch the bottom with either their limbs or their tails. Each side of the tub had a different black and white photo attached to the edge that was meant to be used as a visual cue by the rats. In one region of the tub, a small circular platform that was 8 inches in diameter was placed so that the top was 1 centimeter below the surface of the water. On trial 1 of 6, each rat was placed onto the platform facing the middle of the tub and the visual cues for 10 seconds and allowed to look around the environment without human interference. Then, each rat was placed into the water with its head facing the wall near one of the visual cue pictures. Each rat had 90 seconds to swim to find the platform again. This was repeated 6 times for each rat, placing the rat hind limbs first at slightly different locations around the edge of the tub each time. If the rat did not find the platform on its own before the end of the trial, an experimenter placed the rat on the platform and allowed it to stay there for 10 seconds. For trials 2 through 6, the rat was placed directly into the water and was not given time on the platform before starting the trial. Rats were dried off after their trial ended. Rats were tested in pairs and alternating trials in order to allow rest between each trial. The computer tracking system tracked the rat's movements and pathway of swimming, the amount of time it took each rat to find the platform, the time spent in each quadrant of the tank, and the distance the rat traveled. This test was performed to determine spatial learning in an environment.
The graph above demonstrates the comparison between treatment groups and total distance traveled in the open field test. To test this, we performed a one-way ANOVA that demonstrated a significant statistical significance between the treatments and the total distance traveled by each rat in the open field test. This next graph demonstrates the comparison of the mean number of Sentry enters and the treatment groups. Once again, we performed an ANOVA test to obtain our results, and we found a statistical significant result between the Sentry entries and the treatment groups. For our third graph, we compared the amount of times the different treatment groups went in the open arm in the elevated plus maze. To test this, we once again ran a one-way ANOVA, and we found statistical significance between the treatment groups and the amount of times they went in the open arm in the elevated plus maze. For our fourth graph, we compared the total distance traveled within the elevated plus maze and the different treatment groups. To test this, we once again ran an ANOVA test, and we found no statistical significance between the groups. For our fifth graph, we compare the total amount of time spent by each treatment group in the second quadrant of the Morris water maze. We specifically chose that one because it was the one closest to the platform. To test this, we once again ran a one-way ANOVA, and unfortunately, we did not find any statistical significance between treatment groups and the amount of time spent in the second quadrant. The last graph compares the amount of time that it took each treatment group to find the platform in each of their trials. We predicted that each time it would take them less time to find the platform. And to test this, we once again ran a one-way ANOVA. And unfortunately, we once again did not find any statistical significance that proved this.
Through the 3 tests, we aim to receive answers to our hypotheses of whether or not the rats treated with ketamine will exhibit lower anxiety than any other group. The goal of this experiment was to validate the model by looking for changes in behavior following HCA treatment, and then look to see if ketamine treatment could prevent HCA-induced disorder. Furthermore, after conducting 6 one-way ANOVA tests, it was found that there was significance between total distance and treatment and mean center entries and treatment in the open field test and open arm entries and treatment for the elevated plus maze. These results showed a lower level of anxiety in rats when they were treated with ketamine compared to other treatments. Our results supported our hypothesis, which determined that ketamine would help reduce anxious symptoms. A variety of aspects of our study suggest that our study had strong construct validity. For example, we made sure to be as precise as possible when taking the measurements in our study, since not all rats were handled by the same researcher. Overall, our study had strong internal validity because we made sure that each rat would only be monitored as soon as they were on the specific environment we wanted to measure them in. We also only recorded them for 1 minute each so that they could have an equal amount of data for each rat, and we made sure to also properly label each rat's treatment, that we could properly assign what effect each treatment was having on the rats. Due to the very small amount of rats within our study, our external validity is not the best, since we do not know if the results gained from these rats would be able to be generalizable to other rats and even humans. Throughout the study, there were a few limitations within the procedural aspects of our experiments. For example, during the data analysis, it was noticed that there were different sample sizes in each of the tests. This distinction could cause some variations in the results because there was not an equal number of rats in each treatment group. If performed again, it would be beneficial for all of the treatment groups to have an equal number of subjects consistently and to be very precise when executing the experiment. Also, in a couple of the trials, the data from a few of the rats had to be deleted due to instrumental error. For unexplained reasons, the visual tracking system did not pick up on the movement of the rats. Therefore, it caused inconclusive results for the individual rats. We then had to eliminate the specific data point from our data set. We could have double-checked the equipment to make sure that there would be no error in recording any of the tests. In the future, it would be, if we conducted it again, we would take steps in order to make sure that these limitations were not a part of our study. Overall, the study was very successful, as it was able to prove many of the hypotheses. In this study, it was proven that, in fact, ketamine does have the ability to reduce anxious symptoms, as all of the rats that were treated with the drug had much less anxious behavior than those with the rats that were treated with the saline solution or with those treated with the HCA solution.
Show